Hi, everyone. Welcome to the MRI Together 2022 session on reproducible acquisition practices. My name is Kalina Jordanova, and I am from the National Institute of Standards and Technology in the US, and I'll be the moderator for the session. So today we'll have 4 speakers, each give a talk with short 10-minute discussions after one after the first 2 speakers, and then a second discussion period after. We begin, I just wanted to remind everyone that this session is being broadcasted live and will also be recorded. And the transcripts are available on YouTube if you're interested. Also, I'd like to remind everyone to be aware of the code of conduct for the conference. So during the session, please feel free to ask questions. We'd like people to ask questions in the Q&A box, not in the chat. The Q&A box is the one that has a little question mark on it, and for me, it's on the right side of my screen. So you can go ahead and open it up now if you'd like, so that you can monitor questions as they come in and ask questions of your own. And also, I'd like to encourage everyone to upvote questions that are interesting to them. During the discussion session, we'll generally go through the questions in the order of votes and get through as many as we can. So yeah, feel free to participate. And a last reminder is this is meant to be a discussion, so no question is too simple or silly. Everything is welcome. Yeah, and with that, we'll begin. So the first speaker today is, I'd like to introduce is Xi-Nian Zhou from Beijing Normal University in China. And the talk is titled 3R Brain, a deep resource for reliable, reproducible, and replicable MRI. Unfortunately, Xi-Nian couldn't join due to some VPN and technical issues, but has graciously provided a recording of the presentation. So we're going to begin that now.
I'm very grateful to be invited for MR Together 2022. This is my second time for MR Together conference. And today I will introduce a deep resource for reliable, reproducible, and replicable MRI. We name it 3R Brain. As you can see in the middle of the slides, there's a link for the R Brain. So this is the slides to claim the conflicting interests and also the copyright. And also I'll give the outline of my presentation, including the concept of the R's and the deep phenotyping of our brain and behaviors, and also other 3 pieces of the 3R brains resource, including design and also outcomes, as well as the resources. Finally, I will introduce and acknowledge the working team. First of all, I will give a very brief introduction of the concept of the 3 R's. These concepts are basically raised by the reproducible research, and there are 11 scientific components for a research as illustrated in the left panel of the slides, including population, which is the a complete collection of units for which information is sought. Normally, for all the research, we're talking about a reproducibility, a replicability, and the reliability. The population is fixed. And the second is a question, which is the statement we wish to address in the population of interest. The third is hypothesis, is the proposed explanation of our question that we wish to test. Experimental design is the procedure for sampling and measuring units. Analysis plan is the manner to extract information to answer the question. Data is the manifestation of the experimenter carry out the design. Normally there are many different types of data. And the experimenter is the scientist who will carry out the experimental design. Analyst is the scientist who will carry out the analysis plan as designed. Mode is the manifestation for analyst carrying out the analysis plan. Estimate is the statistical results, estimated by the code. Finally, the claim is the conclusion about the research question, based on the estimates. So for all these 11 elements of the scientific process, we put to the different stages of the scientific research, as demonstrated in the right panel, which show different threats to reproducible science, and also those 11 elements and promising solutions. There are a lot of different threats, as you guys already know. As promising, there are 3 potential solutions as indicated in below, including pre-registration, team science, open science, which is the focus of the conference. So actually in 2017, there are these 3 concepts were formally defined by this consensus study report, and also in this Nature Human Behavior comment. Basically, the reproducibility is reperforming the same analysis with the same code using a different analysis, as as illustrated in the middle of the slides. And the replicability means reperforming the experiment and collecting new data. These elements, including experimental data, analysis code, estimate, and claim, can change during the replicable research. And finally, the reliability actually is a special form of replicability for the question of individual differences with the same analysis and also same set of individuals, which we normally call it a sample. And it means the reliability is the replicability of inter-individual variability across different occurrences. So these are the basic And for reliability, it is a very fundamental concept, to the scientific research. And here I especially highlighted the concept of reliability and validity. So for all the variabilities we observed from individual differences, which can be divided into three different parts, including the disorder or treat-related variability, all those variabilities contentable by other types of individual differences of no interest, and also the random errors. So these weighty and we see altogether, belong to between some variability, the random error we normally call it within subject variability. So from the the portion of individual variability is reliability, which is up for the validity. It means, which is means portion is we normally call it within subject variability. So from the the proportion of inter-individual variability is the reliability, which is upward for the validity. It means which means the proportion of variability of interest to the total variability. So we normally divide the variability levels into different levels of reliability, including slight, fair, moderate, substantial, and almost perfect levels. So why reliability is so important is according to its basic principle or basic theory for experimental design. So from these slides, we can see the sample size is function of both the reliability and effect size given a fixed power, a statistic power. Here, there is a basic statistic rule indicates the effect size we observed is limited by reliability on the true effect size. And given a fixed effect size, studies with more reliable measures need less sample. And for different effect size and given measures with almost perfect reliability means greater than 0.8, but only 3, even for very small size, 300 samples can offer a study 80% power to detect this effect size. So, but for us, for slide reliability means less than 0.2, And we really need big data, more than 1,000 samples to detect these small sizes. So for deep phenotyping, we can say we really need a very large or big data for investigation of those complex or unreliable or small sizes findings. And here, I'll give you an example of brand development, which is based on very reliable measures, like brand volume or other brand morphologic metrics. Here, we used the MR scans, which is really big, from more than thousand subjects, and we can see these are really deep phenotyping of the brain development regarding its morphological measurements. Beyond that, we from another perspective of phenotyping is we need to obtain highly representative individual differences. Means we really need different perspectives of individual differences, including not only focus on development but also focus on different aspects of those factors across micro level, cell level, and also individual level demographics, those factors, and also personal networks and community factors, countries, different regions, as well as those different cultures, which call really efforts on population neuroscience. But according to those big data currently we have, it is not really deep phenotyping those representativeness. So ideally, we want to have extensive sampling from many subjects. So here is another aspect of deep phenotyping according to the representatives of sampling. I introduced the two really big cohorts from China, which can increase the diversity of MR data. The first one is a Chinese imaging genetics project, including more than 10,000 adults in China. And also there's another corresponding project to Human Connectome Project in China. We named it Chinese Human Connectome Project, which employed the identical imaging protocols as the Human Connectome Project. And we can see, based on the outcomes we initially had, indicating almost identical, large-scale population of the human brain resting state functional MRI data. And finally, the 3R brain is really a deep design for reproducible MRI, which includes two parts. The first part, we have 100 participants who repeatedly measured across five scans, which happened at 3 scanners, including one 3 Tesla Siemens scanner, which is Prisma, and another is G750 3 Tesla scanner, and another is Siemens 7 Tesla scanner. Finally, we hope to get 500 scans. And the second part of the design is the picture viewing highly sampled data set. Each subject will view 5,000 pictures in 5 or 10 hours, Alzheimer's sessions. And actually, finally, we got, for the first part of the third operand, we got, 392 scans from 103 participants. This is the actual farmers' sessions. And actually, finally, we got, for the first part of the third operand, we got, 392 scans from 103 participants. This is the actual data we finally have. And then for the second part, we got the 520 scans, which are obtained from the 50 participants who view, these highly dense sample, pictures. So for we already pre-processed and also standardized the data sets of third operand with Bayes and MRQC, pipelines. And so initially, we got some reliability assessments on this on this data. This is the example for cortical thickness, and we can see for intersection reliability, we have really high, or those the cortical thickness are really stable or reliable across all the different networks. But of course, for those primary networks are less reliable. So this is because limited is lower inter-individual differences for these compared to those high order networks. And for interscanner, these this ability is highly quickly reduced into the levels of failure across all the different networks. But of course, for those primary networks are less reliable. So this is because limited is lower inter-individual differences for these compared to those high order networks. But for interscanner, this reliability is highly quickly reduced into the levels of fair reliability, especially for visual central network and the limbic network. And for so for all those data, we will release it at Science Data Bank in early next year, and hopefully this can offer a unique resourceful, I brought together all across the world for those reproducible research. And finally, I want to thank all the members from my main team and contribute to our project. Thank you very much.
Great, thank you very much, Xi-Nian. We will move on to the next speaker. So I'd like to welcome Georg Oeltzschner from Johns Hopkins University in the US, who will talk about data processing and modeling critically affects reproducibility in MRS. So Georg, when you're ready, you're welcome to share your screen.
Yeah, 1 second. I hope this works. Ta-da, ta-da, ta-da. Can you see the screen?
Yes.
Brilliant. Okay. Well, thanks for staying up all night just to see my talk. Let me get this thing out of the way. I can't. No, I can't. How do I get this thing out of the way? All right. We'll just keep it. I forgot to add a disclosure side, so I'll say it's just for everyone that I have absolutely nothing to disclose at this point. I'm going to talk about, other than my title suggested, not just aspects of modeling and processing, because I realized halfway through making the slides that I was in a session called acquisition practices. So I'm going to touch about both things, because I think that both aspects are equally important and plaguing us in the field of MRS. And finally, I want to point out a few, a very, very coarse vision of what we can do as a community to address some of these issues. Now, everybody probably here has a background in imaging, rather, so chances are that you haven't heard of MRS before. So I wanted to give a quick primer. Obviously, when we do proton MR imaging, the vast majority of the signal that we receive is from water, because the body is mostly water and water has 2 protons. But obviously there are other molecules as well that have protons in them, and they are really what MR spectroscopy is all about. And so depending on the chemical environment that these protons are in, they will process at slightly different frequencies. And when we record a spectrum, we plot the intensity of the signal that we receive in our coil over its frequency, really. And we know fairly well, in theory, which peaks represent which compound, and so we get characteristic spectral signatures for each of these molecules, and different molecules that we can detect are involved in different physiological and also pathophysiological processes, some of which are listed here. Now, very, very basically in MRS, what you do is the following. You measure a localized signal, and that can either be in a single voxel fashion, which is mostly what I'm going to be talking about, but also using spectroscopic imaging, where, well, you need frequency to differentiate the different metabolites in the spectrum, so you can't do frequency encoding. Instead, we're relying on phase encoding for spectroscopic imaging. Important to note, there are really, really, really many different ways of doing this, and they're all typically optimized for different purposes and for different applications. It's enormously heterogeneous, which is problematic. But what they all have really in common is that at the end of it, you get one spectrum or more from this acquisition, and they are created by a series of quite complex pre-processing steps. And finally, these spectra are then decomposed into individual metabolite components with different modeling techniques. And most commonly, that is called linear combination modeling, where you approximate the spectrum as a weighted sum, so to say, of known simulated spectral signatures. And during quantification, you attempt to convert the modeling parameters into meaningful units of concentration for these metabolites. And you make all kinds of assumptions about relaxation behavior, for example. And you can already probably see from what I told you, there are so many ways in which you can arrive at the results of these quantitative estimates that heterogeneity and reproducibility are problems. And a very, very good consensus paper on reporting standards has really hammered it home by stating that the translation of clinical practice has really been slowed down and hampered by the lack of standardization in all aspects, and that includes the data acquisition, but also the post-processing, and also a little bit the reporting, like the details of all of these steps are notoriously underreported. And so this is what we're going to touch on during all of these slides today. And one of the key conundrums I frequently find that we face with MRS is that per se, it doesn't generate any images. We generate these spectra and we get quantitative estimates of the metabolites that we see in the spectrum. That is great. We get hard numbers out of this that we can work with. So on the left here, you see an example of the capability of MRS to predict the clinical outcome of neonatal hypoxic ischemia with near perfect sensitivity and specificity if you choose the right quantitative cutoff. But as I've said, these numbers are super susceptible to differences in the way that data are acquired and analyzed. And so we'll talk about this today, about acquisition and modeling and how they affect reproducibility. One good thing to start with is in the field, there is awareness that we have a problem with heterogeneous methods. There was an entire special issue of NMR in biomedicine just over the last 2 years. There's a massive collection of expert consensus articles on practically every relevant spectroscopy issue. If you are looking to get into MRS, these are really a great starting point to understand the state of the field. So let's spend, since we're in an acquisition session, a bit of time on acquisition, actually. And I want to first talk about how reproducible metabolite estimates are between different research sites. So our larger group here at Hopkins, under direction of Richard Eden, has led a large multi-site effort to generate one of the still largest MRS data sets to date. And that is large in MRS world. This is 270 spectra from 25 sites across all 3 vendors. So that tells you we have a little bit of a problem of agglomerating large data sets. What we did acquire was high quality data from really the workhorse sequence of MR spectroscopy, the so-called PRESS sequence. So that is a single voxel localization. You see vendor here coded in color. Different sites are along the x-axis, and the metabolite estimates for N-acetyl aspartate and choline are on the y-axis. So these are 2 very prominent compounds that are detected in spectroscopy. And we find that even if we manage to harmonize the acquisition protocol and the analysis protocols in and of themselves, we're still left with not even so subtle effects of site and vendor due to small remaining differences in implementation and in site different practices. And they are worse for some metabolites than for others. But even if we do a lot on the protocol optimization side, these subtle nuances, they still remain. Now, there are ways to correct for this after the fact. So Tiffany Bell and Ashley Harris, they took this big data set and they wanted to investigate whether there is a way to remove this between site and between vendor variance. And they used an algorithm, Combat, which is combating batch, I believe. So just an approach that is, I think, usually used in genetics or gene expression microarray data, something like that, has since been successfully applied to fMRI data as well. And it shows, well, there are ways to remove this variance that is associated with site and vendor. Obviously, it's going to be more generalizable the more data the algorithm has to play with. So this is an instance where we feel like we need to do a little bit more in terms of acquiring large data sets. Obviously, you also run the risk of removing site-specific variance that isn't associated with acquisition, but let's say one associated with specific populations at each site. So ideally, we can find ways to have smaller effects of vendor and site in the first place. The data that I just showed you before, I told you were acquired with the product spectroscopy sequences that are provided by the 3 major vendors, right? GE, Philips, and Siemens. they've had these basic sequences in their stock for decades now. But as it turns out, they are sufficiently different in the implementation, in sequence timing, in slice profiles, water suppression, shimming, and all these are quite likely the culprits for the site-to-site and vendor-to-vendor differences that we observe. Fortunately, there have been recent efforts to address and to harmonize the acquisition across vendors. So here is a great sequence that was a collaboration between a whole host of really excellent sequence developers, following a expert consensus recommendation for semi-laser localization, which offers a few advantages over this workhorse press sequence. This is still relatively new, and we don't have that much data, to be honest, but from the few volunteers that were scanned in this particular study, in the original paper, there seemed to be quite good consistency. As you see here on the right-hand side, the bar plots show the consistency of the metabolite estimates between scanners and between vendors. Variants appear to be quite comparable too. So hopefully this kind of work will feature future improved uptake in the community. Obviously, another thing that I just touched upon is reporting, which has been a huge problem in spectroscopy for a long time. If you want to ensure reproducibility and repeatability, you need to provide a lot of details, not about the acquisition, but also the analysis. Fortunately, in the course of this wave of consensus papers, experts have compiled a minimum reporting standards checklist. I'm not going to go through the details this much, but it's just very useful to have as a checklist for reporting. So, for example, what you saw before: hardware details, field strength, type of scanner, information about the acquisition itself. So that would be pulse sequence, voxel size, echo time, motion correction methods, and all that. But also really, really, really important, report in detail how data are analyzed. So which software is being used, which analysis steps, how quantification was being performed. And finally, some quality assessment metrics. So showing representative spectra and reporting QA. So these are hopefully minimum standards that will catch on and help us in the future to compare well, the methods themselves, but also their outcome with each other. Another source of variability that is probably quite substantial is the position where data is being acquired from, which is especially true since MRS is usually, well, not done in this whole brain fashion that we use for imaging, but rather by placing just a single region of interest, which is usually just done manually, right? So by scanner operators or by techs. And there is obviously some operator bias there, and even the same operator won't perfectly nail the same voxel placement every single time or across individuals. So people have proposed to do automated atlas-based voxel placement. And here the ROI is essentially defined in MNI space or some other standardized space and determines then the coordinates for voxel placement after transforming the individual T1 into that space while the measurement is taking place. And it does appear that this does substantially increase the degree of overlap between regions of interest, and that is both true between individuals, but also within individuals, so between sessions in the same individual. So this could be useful, but again, the uptake is the problem. These things are not featured in product yet. These are all essentially local sequence developments that are distributed through patches or through CP agreements. So far, because we're short on time, I wanted also to talk about the analysis side of things, and these two are kind of intricately linked, like even more so, I believe, than in imaging, because we're looking at extremely small signals and we want to account for all the idiosyncrasies of the small of these different sequences. Because even if you manage to really perfectly harmonize the acquisitions between sites, it matters to a really frightening degree how data are being modeled. The way we extract quantitative information from spectra is by fitting them with quite a complex procedure, which is called linear combination modeling. And there are many different algorithms and implementations of how this can be done. Many different software packages. The most prominent one, LC model, that you might even have heard about if you're not a spectroscopist. That was a black box commercial product for a really long time. Arguably, that hasn't helped much with transparency. There's a recent wave of open source solutions, including Osprey, which, full disclosure, is one of our developments in our group at Hopkins. But we're generally very interested in just investigating how much of a difference does it really make, what kind of analysis you're using. So we took, again, these 270 spectra that I was talking about earlier, and we just fitted them with the Osprey software, with this LC model, black box workhorse, and the Tarquin software, which is another tool that is quite frequently used. And it shows that even for the strongest signals in the spectrum, which is, again, N-acetyl aspartate and choline, but also myo-inositol and glutamate and glutamine, we found there are substantial differences between the tools in terms of the mean estimates. So here visualized in different colors. This maybe would be acceptable if it was just an offset, like a systemic difference that just shifts every data set in the same way between tools. But the cross-tool correlations turn out to be relatively poor as well. And that is really unfortunate, right? You want these correlation plots between tools on a perfect line along the main diagonal, right? That would mean they would agree, but instead you have these large point clouds. So there's not just this systematic bias for each tool, but there's actual disagreement between the tools. And it means that the choice of tool introduces substantial variance into the quantitative results, which is problematic. In a variance partition analysis, we found that depending on which metabolite you're looking at, the choice of algorithm can account for up to 60% of variance. And that is just a huge problem. And already there's a lot of potential for improvement. What's problematic with looking just at large numbers of in vivo data sets is that obviously you lack a ground truth, right? So you rely on your modeling algorithm to pull out the right numbers for you, but it's just not really telling you whether these numbers are right. So what we're fortunately pretty good at is doing simulations of what our metabolite signals actually look like. And we can create synthetic spectra that we can drag through our fitting algorithms as well. This is a relatively recent development in MRS, hasn't been done a lot. But the ISMRM spectroscopy study group has organized a fitting challenge with artificial data, varying just in the relative contributions of these metabolites, also in spectral quality. And the challenge was essentially make a pipeline, whatever you want to use, fit this with a software of your choice, and see how close you get to the original ground truth values. There were 26 teams that participated, and they used a whole bunch of different packages and settings. But, well, what can you say? That the fits to the data, they all look pretty good. So the model that was being used, regardless of the software, managed to approximate the data really well. The problem was that quantitatively, these solutions didn't agree at all, not with the ground truth. So what you see here on the right-hand side is the percentage difference between the measured concentration and the ground truth for different submissions. So the submissions are on the x-axis. And even the most widely used package, this LC model software, would easily bring in between 10 and 20% of bias. So if disagreement between measurement and ground truth. They also didn't really well agree with each other. There were huge differences depending on what kind of settings were used. So this is in other ways even more concerning, but at least I think it's starting to take hold that we are starting to appreciate the fact that variability is there and that we need to work on validating our modeling results much better. And obviously, to kind of close the loop to a lot of the other methods that are being touched upon, it's something that other neuroimaging disciplines obviously encounter as well. For example, here on the right-hand side, this famous fMRI paper in Nature. The abstracts of these two papers really map well onto each other. So I think it's a nice reminder that we're facing similar problems. Key point is the conclusions drawn from data sets really vary substantially depending on the way that you analyze and model them. Now, finally, just let me briefly, in the remaining 2 or 3 minutes, touch on what can we do about all of these things. And the point that I maintain is that for the longest time, MRS has a problem with his infrastructure, really. So I recently wrote a grant application for a pathway to make MRS more useful and more reproducible and scalable. Now, the first thing is kind of a given almost. We're aware of the issues of quantitative analysis, so I'll assume that we just managed to implement the methodological consensus laid out in all of these papers, right? That will take time. I hope we ultimately get to somewhat standardized agreeable acquisition protocols, and just agree on ways to analyze the data. What we really need, I think, is an infrastructure that then allows us to aggregate a lot of normative data, a lot of pathological data as well, that allows us to deploy spectroscopy at scale in multi-site clinical trials, because we need to be able to be generating more robust evidence that goes just beyond a few subjects per study. One thing I'm really, really proud of what we have achieved over the pandemic period was that we founded an initiative to transform the MRS community in particular towards open science practices. So we have a standing study group committee now that creates open source tools and resources just to facilitate this transition towards more collaboration, towards more aggregation of data. And we are also engaging with a lot of junior researchers, which is really, really nice and has provided a great deal of motivation. Our web portal, the MRS Hub, has become a really, really lively, nice gathering place for the community where they can exchange support and ideas and data and analysis tools as well. And this really makes me hopeful that as a community, we can take the steps that are necessary towards combating these issues. One thing that has come out of this community effort already is that we're looking at next generation MRS infrastructure. And one of the prerequisites for that is that we learn to speak the same language. Like there were vendors have traditionally developed all their little kind of like in-house spectroscopy data formats, because spectroscopy doesn't really lend itself to be used with DICOM, which has been an absolute nightmare for people who want to write software or who want to share their tools. And thanks to Will Clark, who's presenting, I believe, in another session. So if you're interested into MRS, look him up and his fantastic work. Thanks to him and Martin Wilson, we have let this community organize a nifty MRS standard, which is hopefully going to become the standard to replace all these vendor-specific formats and make it easier to not just develop software, but also to exchange data and just aggregate and agglomerate larger data sets and to exchange them between sites. We are thinking of making huge data sets available to the community just without having to worry about the formats. And once we have Nifty, the cool thing is it's a short step then to think about how can we incorporate spectroscopy into the larger BITS infrastructure. That is also underway. There's currently a BITS extension proposal under review, so hopefully this will be out hopefully within the next couple of months, and it will be a great way for us not to deploy MRS at scale, but also have an easier way of integrating it with all the other fantastic modalities that everybody else is working on. And finally, one of our collaborators, Alex Lin, in Boston at Brigham and Women's, he's pioneering some ways to aggregate pathological data, normative data, in a way that has just been unprecedented for spectroscopy before. We hope that can also serve as a basis for methods development in the future, but it's just a great first step to see, and all of these efforts have directly or indirectly been born out of our community efforts. So just to wrap it up, I still think for clinical purposes and for research purposes, we still have a lot of potential of MRS that we're not really mining, and largely that has been because we have failed in the past of really creating an infrastructure that allows us to do somewhat similar things and then exchange the data and the methods that we've been using. And hopefully the next generation infrastructure that we're currently creating is going to be helpful in addressing the issues of acquisition and analysis that I've outlined before. I think I'm like slightly over time. I hope you don't mind that. We can cut into the discussion. And these are my fabulous mentors and postdocs and collaborators and friends and the funding. And with that, thank you very much for watching, listening, and happy to take any questions that might come up.
Thank you very much. That was a great talk.
Oops.
Well, let's see. We're still waiting for some questions to come in. So everyone, please feel free to ask them.
Yeah, it's MRS bits cannot come soon enough. It's true. It's on the way. There's a an almost beta implementation already. If you are familiar with Bitcoin, the software. I think John, you're at Rapout. Are you still, or you were? I'm not even quite sure. But there's someone who has been creating the Bitcoin software, which is just this all-in-one converter for DICOMs into bits, and he has implemented a plugin that does kind of a preliminary conversion for spectroscopy data into the bits format already. Like I've been looking into that over the last couple of weeks, and it's still a bit like rocky, and it doesn't support all the like vast array of spectroscopy methods and formats that are out there, but we're really getting there. Like this has been, in the last 2 years or so, we've made major progress, and also particularly thanks to like Will's progress on the Nifty MRS tool. So we're getting there.
Yeah, no, that I'm looking forward to it. But yeah, that's also my neighbors in the next office, so I'll knock at the door for that.
Are you next door to Marcel?
Yes.
Oh, say hi to him.
So yeah, I had a question actually. I can just ask it. So, what about the animal stuff? Like a lot of the spectroscopy started with animal stuff and Bruker implementations. I checked your tool today and Osprey, and then there's no Bruker support yet. Is this something not enough?
Yeah,
not enough of a market, or?
Mostly because we've been historically building it from our own purposes, but there have been people who are reaching out about animal data for sure. Like the good thing is that for most Bruker data, Will Clark has this converter into Nifty MRS format, and we can read in the Nifty MRS format. So I think that would be a way to get it in there. And yeah, I would envision it's not much of a problem to really make it work. I think most of the like some of the pre-processing routines that we have in our own software, they're a little bit human specific, but as long as you're like staying in the brain, then most things should work. And the modeling is kind of is going to be pretty much the same whether you're doing it in the mouse or in the rat or in the human. This is all fairly doable. And I know a good amount of folks who do preclinical work who we can always tap into for further resources. So it's not something that we actively do, but if somebody comes to us and says we desperately need this and we can't imagine anybody else to do it, then we'll find ways to do that, right? Like it's not a fundamental like obstacle.
We have a question from Sophie. You're welcome to join if you'd like, Sophie, and ask, or I can ask. The question is, what are the largest challenges for open and reproducible MRS methods, and how are these challenges different from MRI?
Fabulous question. Okay, cool. Great. Thanks for writing it out. It touches on several different things. I think that one thing that I've touched upon, I think, is really validation. I think because the numbers that we're pulling out of these spectra, we can never really be sure that these are true numbers, right? Like it all comes down to modeling a bunch of lines, and there are so many algorithms that can do that, that we have not done a great job yet assessing that we're doing that right. And there, I think synthetic data is really going to help us massively, because we have become pretty damn good at creating artificial spectra, and a lot of people are creating artificial spectra at this time already to train deep learning networks on doing the same thing. So I think there's a lot of work still to do. I'm not sure we'll arrive at an ultimate perfect answer that will, for all scenarios and for all kinds of spectra, will give us the correct answer, but I think it'll tell us a little bit about where we can make these algorithms better. So that is one thing that I believe on the modeling side we can make a lot of progress. And the other thing really is to me infrastructure, because as I said in the beginning, the acquisition is terribly inhomogeneous, and it comes down to, like, if you ask 50 spectroscopists what is the best way to acquire your data, tell me your local acquisition protocol, you'll get 50 different protocols. So we're only slowly getting towards things like almost like white paper, like white papers that tell us about the best acquisition practices. I think the expert consensus papers were like halfway there. But yeah, like protocols are one thing, and then even acquisition sequences are the other. Like for whatever kind of purpose you choose, there might be a slightly more specific MRS sequence to do that. And like people use different echo times if they want to specifically focus on one particular metabolite. People use what's called spectral editing sequences that isolates very small signals like GABA, for example, or lactate. And everybody might have their own local implementations of all of this. So even on the acquisition side, everybody's almost doing different things. And then on the analysis side, that potentiates, like everybody uses even more different pipelines on the analysis and modeling side. And that has been, I think, a huge roadblock, because everybody comes up with slightly different numbers, which depend on the specific acquisition analysis set that they're using. And whenever you want to transform like a specific like cutoff value for diagnostic purposes or something, you essentially have to establish that cutoff value for every different site again with local control populations. And that is where I think that normative data could help us a little more and help local sites benchmark their results against established normative data. And in order to solve all this, we just need infrastructure like better exchange of data, which is where the bits formats come in, and hopefully large repositories as well.
Okay. Yeah, it seems like a hard problem. We have less than a minute left, but there is another question.
Yeah, comprehensive is a very nice word for saying that was way too long.
Yeah, so real quick, in less than a minute, are there considerations for using the updated Kiba profile template for capturing and communicating standard MRS standards?
All right, I'm going to out myself as a total idiot, but what is QIBA? Is that like quantitative imaging biomarker?
Yeah, exactly.
Is that an NIH framework? Probably. I'm glad you're bringing that to my attention. Probably is very embarrassing that I was not aware of it, but
Anonymous brought it there was a question from the crowd, by the way.
I want to say yes, and I think the problem with doing that is really reaching community consensus. Like even if you, and then implementing the consensus. Like even if you find kind of the lowest common denominator that everybody can agree on, I guarantee you that some sites will still run their own local protocol because they think it's better than that. So it really takes commitment, I think, from each community site member to just say, all right, we're just going to stick to whatever we agree on, because that is the only way we can make our things comparable between sites. But I like the idea, and I will take a note on that and muse a little bit on it.
Great, thanks. Okay, I think we should move on to the third talk. Thank you very much. So for anyone who's joined, just a reminder that you're welcome to ask questions in the Q&A box. People have been doing it, so you've probably seen that. Okay, so the next talk The next speaker is Todor Karaulanov from Caliber MRI in the US. And the title of the talk is Fully Integrated Standardization Platform to Advance Quantitative MR Imaging. So yeah, I see Todor. Yeah, feel free to share your screen whenever you're ready.
Okay. Hello, do you see my screen?
Yes.
Okay. So hi. So I'm going to discuss our approach on on a fully integrated platform to advance quantitative MRI imaging, especially in one of its modality, DWI profile of KIBA, implemented in I-SPY 2 clinical trial. So this work was done in collaboration with many partners and funded under NIH NCI SBR grants called the Quality Control Program for Quantitative MRI Data Acquisition and Analysis in a Multi-Site Clinical Trial. So the agenda is, we'll start with a little bit of introduction, then we're going to discuss the need for the standardization in quantitative MRI. I think that's a no-brainer. Then we're going to talk very shortly about our integrated QA platform and how actually we standardized quantitative imaging in practice. As I said, that's going to be on the basis of I-SPY 2 clinical trial. And then we're going to talk about a little bit about KIBA conformance, and that's KIBA is RSNA organization. It's Quantitative Imaging Biomarker Alliance, and we're going to discuss a little bit the DWI profile. So in the introduction, so the need of quantitation in MRI or QMRI, why we do the extra mile? Well, first of all, we kind of know that there are at least 2 books on the subject, maybe even more than that nowadays. And that's a good reason, but in reality, if we think carefully, so most of the technologies in the medical field are quantitative in nature. MRI and some other imaging modalities are the only ones that does not really use quantitation, and the reasons are quite complex, and they range from purely historical nature to cost effectiveness. And nowadays, however, the cost effectiveness of quantitative methods in MRI has been mostly resolved, but still not implemented in practice. So what are the important quantitative MRI modalities? Well, the major one is reflexometry in terms of variety of techniques. So we're talking about the T1 and T2 mapping. Then we have the diffusivity, apparent diffusion coefficient, high order diffusion moments like kurtosis. We can think of more than one-dimensional diffusivity in terms of diffusion tensor imaging. Then we have intrinsic tissue magnetism as a quantitative technique. That's the QSM, quantitative susceptibility mapping. And we have another area where we can call them MRI transfers, like the CEST imaging, magnetization transfer in general. and others. So the need for standardization in this quantitative MRI, well, as any quantitative technique, we have to have a ruler, and we have to have a baseline, and we have to have a standard. And here is a very nice paper with great language saying that the quantitative MRI has the potential to make a great clinical impact, both in diagnostics, early detection of diseases, and then even replacing a biopsy, and provide a clear differentiation of disease states. And the interesting part here says it's clear numeric differentiation, so very different from what grayscale imaging provides. And it also says that it requires reference materials and reference objects. So the recipe, we can summarize it as following. You just repeat the following cycle. You start with some reference objects that are very well characterized. They have, for example, if it's a relaxometry phantom, that's going to have a T1 and T2 characteristic of very simple solutions that are very well known and preferably measured by NIST, even better if they're traceable to NIST. And then you can continue and have a proton density arrays where the proton density is known very well. Then we can go on and move to apparent diffusion coefficients and so on and so forth. So once we have this reference object, or it's actually many reference objects in order to cover the entire range, entire physiological range, including all the pathologies. So then we have to load these objects in a scanner and run a specific acquisition protocol that, most importantly, is fixed and standardized for all the scanners that are tested in a study. So, for example, here we see that that's the inversion recovery series. So we're providing T1 maps, and the sequences are adjusted for GE, Philips, and Siemens. Essentially, we can think that is the same sequence, but in reality, we know that it's not the case. There are differences between manufacturers that sometimes are quite significant. So we adjust the sequences between the different manufacturers and between the different scanners. So, and we follow the same protocol. And at the end of the day, after we run the protocol, we analyze the data. That's going to be very important, what kind of software and what kind of conventions we're going to use to analyze the data. Then we create a table of biases. And the bias is the MRI measured quantitative parameters versus the NIST NMR measured values, or NIST certified values if possible. And then we take actions if these biases goes beyond, or they exceed predefined values. For example, if we're measuring apparent diffusion coefficient, and if we put a threshold of 10%, every time when these biases are more than plus minus 10%, this should raise an alarm, and we have to take some action and figure out why the scanner is performing in such a way to produce more than 10% bias. In some imaging modalities, perhaps 10% is too much, so we have to adjust our threshold. So what we have done at the company is to create all kinds of reference objects depending on the imaging modality, if we want to be standard, and actually physiology and anatomical features. So, for example, that's the well-known NIST phantom, and here we have breast. On the right, we have a breast phantom. So their names tells us that the first one's going to be actually for is head phantom by the look, and of course breast phantom is standardizing breast with respect to T1 and ADC properties. And then somehow we have to couple this the results from the scanner to a software, to a single software, and this software could be open source, could be closed source. So here our software is a closed source, but we have open source component that is actually available. That's the Python core is available as open source. And this way, having the same reference object, the reference object and a software that works together and that are tested together many times is a recipe to perform this accurate and repeatable and to obtain this accurate, repeatable and reproducible results, and to provide better quantitative imaging, and of course to lower the cost this way. How it works in practice? Well, let's consider the I-SPY 2 clinical trial that we are part of it. So that's a collaboration between the I-SPY 2 imaging working group. That is Nola Hilton at UCSF and Caliber MRI. This is, as I said in the beginning, we have a common SBR grant from NIH to produce this platform and apply it in the I-SPY 2 clinical trial. So the I-SPY trial is a NAS trial, so it's a neoadjuvant systemic therapy trial where different drugs are entering in the trial, and we have usually patients that are on more than one drug, and there should be a decision when the drug has to change. So a biomarker is usually used to figure out if a pathological complete response or any response to a given chemotherapy is produced. So, and how we do the realization of the standardization, here is the phantom, and as we described before, we have for now we have 7 actually sites. Here I present data from only 4 sites. So for about 7 months, here's the data from about 7 months or so, and we run between these 4 sites this phantom every month, and we produce the DWI images. We extract the apparent diffusion coefficients. Everything is totally automated. There is not much user input, and that's a good thing. And at the end of the day, we are producing the results. This is the ADC bias for all 4 different sites, A, B, C, and D, with time. So these are the average values over several months. Now we have data that's going to be presented next year at the ISMRM that spans about 16 months, and we have 6 sites. This data was actually presented this year at ISMRM, and you see that the temperature uncertainty, that's the NIST value. So this is the deviation from NIST value of the ADC of different vials that we measured, and the temperature uncertainty is the gray. So everything within the temperature uncertainty should be okay, but we see that we have outliers. Some are on the border, some are outside, almost up to 10%, and some values exceed 20%, and their uncertainty is larger for the bias, and that's not acceptable. So we have to take actions and figure out what went wrong with this particular site. We see that's the site B, and of course site C. We see for some apparent diffusion coefficient, for example, 10% means that this is the active chemical, which means this is closer to water. So the scanner is performing very well. But when you have a pathology that is this presented by in the red, these are the different mimics represented in different pathologies, you're seeing a deviation at about 7-8%, and that's perhaps too much. So, so the Kiba conformance. So what we were implementing in the I-SPY 2 is our first application of. We are running actually the Kiba profile, although we are not running Kiba conformance, but we are following all the steps except one. That's perhaps I'm going to mention. And so this Kiba conformance that we are trying to implement into I-SPY, and we're also offering to external organizations that we can certify them, so we are following the Kiba DWI profile and trying to achieve useful level of performance for a given biomarker. The biomarker here is diffusion. And the profile includes claims related to the ADC, to disease presence, of course. For example, one such claim could be that a measured change in ADC of a brain lesion of 11% or larger indicates that the true change has occurred within 95% confidence. So if our error in determination of ADC is of order of a 10% or so, it's going to be very hard to differentiate longitudinally and across sites any real changes in the ADC in the cohort and within a single, of course, patient longitudinally. Yes, so, and we are trying also to establish and implement this into the I-SPY 2. This is, for example, the phantom that is used. This is diffusion phantom for this profile. And I would say I'm going to stop for now, since there may be some questions. Thank you.
Thank you. That was great. Before we go into the questions and the next talk, everyone feel free to write down your questions now, and then we'll go to the fourth talk, and then we'll discuss both the third and fourth presentations at the end. Thank you very much. Okay, so I'll introduce the final speaker for this session, Joe Grandjean from Radboud University in the Netherlands. The talk is titled Approaches for Standardizing Preclinical Functional MRI. Go for it.
Thank you very much for the introduction. You should be seeing my screen now.
Yep, you see it.
Perfect. I'm very happy to be here. Thank you so much for inviting me. I'm also the last in the session, so I'm also aware that what I do is very niche. So in case this is really not applicable to you, I've put everywhere as much as I could paintings. I was very much inspired while preparing my slides today by the work from a Swiss painter, Ferdinand Hödler. So, you know, if the talk does not apply to you, at least please just enjoy that. So what I want to talk about is really about the work that we've made as a community to make rodent fMRI better for everyone. And this is not really just one person's work, but it's really the work of many people working together. And spoilers, actually reaching high-level goals. I give you the outcome already at the right now. We've made rat functional MRI 50% better for everyone in the community, and I'll explain to you what's the approach that we did to get there. But first is the motivation, and I think the drive for everyone especially now working in neuroscience, is try to understand brain disorders. This is a challenge for our time. The numbers are huge, and I'm not going to repeat them. But I want to show you two major landmark studies, which are the inspiration for me for my own work. And of course, yes, this is about patient data, because that's where it always is. What we have on the left-hand side is a classic study from Jevet in 1997 using nuclear imaging, where they show hypometabolism in groups of patients with depression compared to control, showing a clear sign of hypometabolism in the prefrontal cortex in these participants. And this is important because it showed that depression was anchored into a biological phenotype that you could detect into the brain. On the right-hand side, I show the study by Filippini. And what they did is they imaged healthy carrier for risk allele to develop Alzheimer's disease later in life. So these are participants who are in the 30s, who are healthy, but who have a greater chances of developing Alzheimer's disease. And this is resting state fMRI, showing here one of the so-called network called the differ mode network. And this is a contrast image where they contrast the carrier for the risk allele versus controls. And they do show that already before the disease appears, there are visible changes in the brain that you can detect with brain function. So these are my personal source of motivation. I just want to share it to you because I think it sort of helps contextualize. But it's not just inspiration for me. I think the work in human neuroimaging has been stellar and has inspired a whole community to come together and to generate resources that are now just in the common space. We have the Human Connectome projects, which is one of the mostly used data set out there with a thousand MRI scans, the UK Biobank. And just a few minutes earlier, we heard about the effort in China to also aggregate large MRI data sets. But the story I want to tell you is quite different. I'm talking about animal stuff, and this is very niche. This is we went through with my students and we counted the number of publications where they performed animal functional MRI. We see that rat is the most common species used, followed by non-human primates. Eventually mice come, and then we have other further niche animals. But what that plot shows you is that on a good year, we can best hope to have maybe 100 or so papers on the wide topic of fMRI in small animals. So it's nothing with the kind of scale that we find and we expect in the human neuroimaging. So I'm not, claiming to change the world with the work that we do, but I will try to show you that we've made a meaningful contribution nonetheless. Still, again, to show the relevance of animal work, I just take two snapshots from two of my recent work, showcasing here how we've applied our research to study depression, where we show on the top left part a heat map that shows a literature meta-analysis for the search term depression that really shows that on average, studies tend to report areas in the brain associated with depressions in very localized parts of the brain, in particular the cingulate cortex, which you find here along this midline. And in work that we've done with an animal model of so-called depression, we do find that the brain state, the main brain state that is affected in these animals relative to control, also features an analogous region called the cingulate cortex. And to look at Alzheimer's disease, here we performed the same literature meta-analysis, but this time for tauopathy, which is a pathological aspect of Alzheimer's disease. And we do see ventral networks being highlighted more often than not in associations with this pathology. And by putting a mouse model that expressed the same pathology, we also do find functional deficit in the corresponding areas in the mouse brain. So this illustrates that we have the potential to make a difference, because once we've sort of identified homologous markers, then we can go into the depth and try to disentangle what it actually means to have functional connectivity deficit in terms of biology. And we do that very much in our paper, so I encourage you to have a look eventually. But fMRI really is challenging. It's challenging also in humans because we often work at the thresholds of detection. It's an indirect physiological measurements. It's confounded by noise sources. It has a complex pre-processing. No offense to the people working on MRS, and I know MRS is complicated, but I like to put forward that fMRI is even crazier, if I can say, and the interpretation still remain difficult. And again, if I'm allowed to contrast to spectroscopy, right, where we have, for instance, specific peak that corresponds to metabolites that we can relate, no one is yet capable of telling what a decrease in functional connectivity really means in terms of biology. Now, when we turn to rodents, this is made even more complicated by the fact that we have to work extra hard to maintain proper physiology. We have a huge debate in our community between awake versus anesthetized imaging, with stress that might be involved onto the system. We have even further stress limitations with signal to noise because we image smaller objects. And if we talk of standardizations, then we even face the fact that we work with a larger range of magnetic fields than we do commonly find in humans, but also with people just getting used to building their own coils and their own platforms, whereas in the human side, you would tend to buy coils or platforms from established vendors. So what we did in that study is we aggregated 82 resting state fMRI data sets in the rat. That's the work not of myself, but it involves way over 200 collaborators from 50 research centers distributed across 20 countries, and from MRI scanners ranging from 3 Tesla to 17 Tesla. And again, just that you sort of contextualize the work that we've done, this is the map of the sites where the different people provided data or where we had contributors. I have to acknowledge this is very European-centric, and to some extent we have the East Coast in the US. I've been terrible, I believe, at reaching out to people in other communities in the US, but also in Asia. And if I haven't contacted you, or if we haven't made contact yet with you, or vice versa, please do feel free to reach out, and I'd like to include you in future studies that we're planning. So we've aggregated these data sets, and I think the first nice thing to look about it is just how diverse the rat data sets that we collected were. And we find the usual biases with the fact that researchers, unfortunately, keep on having a sex bias when they image, with males being found more often than females. When it comes to rats, people image with many different strains of rats, so they are slightly genetically different, with different also physiology and behavior. People also handle the rats differently with different anesthesia protocols, and also they use different field strength. Just to give you a sense of how diverse the data set that we've aggregated was, but just also for you to visualize sort of the challenge involved in processing that is just how different the MRI images look like from one data set to the other. And here I'm being nice in this representative figure showing one slice across the rat brain in 3 different scans, where we do have really different patterns of image distortions. But again, the images that I've selected still represent, or we can tell that there's a brain in it, but of course people have sent me data that also looks completely off as well. But where I want to get at is the greatest advances in the work that we've done as a community, I believe, is agree on what we want to look at into the brain. And for every discussions that we've had before in these sessions, then I think this agreement, getting to this agreement is really, that's where the problem is. People don't necessarily look for the same stuff in the data, and getting people together with the same idea was the biggest challenge, and I think that we've pulled it off tells a lot. When it comes to resting state functional connectivity with MRI, it will be difficult to get people to agree on what is the ultimate metric to look at. We've proposed that there are 2 properties that maybe I think 90% of the community would agree with. The first one is a temporal property, which is characterized by an absence of correlation between task positive and so-called task negative network. This is what is illustrated here in this classic figure by Fox and colleagues, where they show that 2 regions in the same network called differ mode network have time series that correlate with one another, whereas in a different network that would be activated during a task, the signal is, in that particular case, anti-correlated. So this is something we can work with. The other thing is a spatial property, and it's coming from the observations also in humans, that most of the networks that are observed have a contralateral homotopic pattern that we can see, and this is really symbolized here by this viral tracer injection in a mouse from the Allen Brain Institute data sets, where they injected a tracer into the sensory cortex. And what you see is that the neurons don't project randomly into the brain, but where they would tend to project is the contralateral homotopic area. So based on these 2 properties, we've made this plot, which I think is the most important plot here, where on the x-axis we show correlations between 2 regions where we would expect biologically to have correlations. So in that case, the sensory cortex, left and right, and the value is expressed as Pearson correlations. So the correlation between the signal extracted from one region here to the other region on the other hemisphere. And on the y-axis, we show the correlation between the sensory cortex and an area where we do not expect to have correlations. So that's just to explain how this plot is. And then this plot we further divide into different areas. The first one being here at the center, which indicates the presence of scans, each indicated by a single dot. And this is one such example where there's no pattern of functional connectivity that we could detect with the pre-processing that we've done. And this is about 30% of the material that was supplied to me. So 30% of the scans that I received from my colleagues and collaborators were quote unquote empty. Another, about a third of the scans that we received either had patterns that were spurious or non-specific, where we would see some patterns of correlations, but they don't make sense with our biological expectations. In total, finally, we only had about 37% of the material that was provided to me where we could find patterns that would kind of correspond to our biological expectations, which is represented by this scan here. a little bit of statistics and a lot of discussions with the community that followed these observations. And we came up with a new protocol for acquisition that includes a minimal MRI protocol, as well as an anesthesia protocol that is quite detailed, that we've proposed as a standard way to acquire resting state fMRI in rats. The protocol has been designed to be minimalistic, and I want it to be, the benchmark was that the protocol should be able to run on scanners that is 20 years old and low field. So if you have something modern, then you should absolutely be able to run that protocol. I'm not going into great depth into that, but the outcome from that was that when people went back with that protocol to the scanner and acquired a new data set with that new protocol, we were able to improve the number of scans in that good part of the plot by 50%. So now we're about 67% of good scans on average in a data set, which is an improvement from the 37% that we had earlier. So in summary, what we've done is a democratization of rodent fMRI. And I want to highlight, before I finish, how we've also achieved that through open science. And so we've used the classical tools for open science. So the study was pre-registered. My collaborators at McGill University developed a software toolbox for pre-processing that's extremely robust. For people who are not familiar with data analysis, we also provide all the code to replicate the entire analysis that we performed. And we also provide a new vocabulary to express how we do quality control in fMRI data in small animals, which is, for instance, represented here. So altogether, it's not just that we've made a protocol that's 50% better, but we also provide the entire methods around it to help everybody achieve this. And of course, this is not one person's work, but it's the work of many people put together, and I have to thank them. But in particular, I have to thank my collaborators at McGill University from the team of Mala Chakravarti, who've made fantastic work with the software support. And then that's it. So again, if you are into the animal MRI sphere and you have interest in functional connectivity, and I haven't contacted you yet, but please do get in touch with me, and I'd be happy to chat anytime.
Great, thank you very much. We have about 20 minutes for discussion. So feel free to go over to the Q&A section and post questions if you have any, or yeah, we can discuss here as well. I posted one that I can say. It's for Todor. So yeah, great talk. I'm wondering how often there are software upgrades for each of the sites, and if you need to restart the averaging every time there's a change. Like basically, how much does the software affect the measurements, and if you have a feel for that, or if the measurements are pretty stable even with upgrades?
Sure. So both software and hardware upgrades, well, one is update, the other is upgrade, are affecting, could affect the quantitative nature of the MRI output. And it has been observed many times that after a software upgrade, you can have a significant difference in the reading. And I guess that has something to do with the way a particular sequence is executed. And there, in some cases, the difference between before and after the upgrade could be up to 50% in terms of absolute values. So, and that's huge. Yes, and so we always recommend that after software updates or hardware upgrade, before that and after that, you should run, reference objects and quantitative sequences if you're doing any QMRI studies.
And is the idea kind of, if the results are, what do you do if they aren't the same? I mean, this is a problem we have as well. Like, what do you do if you run those reference scans before and after and they aren't the same?
So that's a good question. And usually you have to go back and refer to the OEM manufacturer and ask for support, because you know that you should not have such a large biases. It's almost going to use render the scanner useless in terms of QMRI sequences. And who knows, even running other sequences, they also could be affected.
Yep. Okay, Sophie has a question for Joe. Fantastic talk and work. How much have you looked into the scans in your top quadrants? Are they bad scans because of known technical reasons, or could it actually be something new and some type of new and unexpected biology?
Yeah, it's contentious. My belief is it does not account to proper biology or proper expectations. As far as we know from, tens of thousands of MRI scans in humans. So I'd like to believe that these are artifacts, but people will argue for their own data set and pretend that it could be something else. So I like to be non-dogmatic. It's not what I would expect, but maybe it's there.
Okay, fair. Another question for Joe. Are there any unique challenges for open science and preclinical MRI in terms of data sharing? I don't work in the field, so this may be silly. Yeah. But do you deal with data privacy concerns?
Yeah, no, that's the thing. I mean, in theory, animal work should be so much easy to share. And like, it's a privilege to be allowed to work with animals. We have to work really hard to get licenses and everything for it, so that we should really make the best out of it and share the data. But I think people have still that very close mentality about sharing animal data. For me, the advantage is like the whole work that's been done by the human community with Open Neuro and all these tools that just makes it so simple to share data nowadays. And so we are indebted to the human MRI community in a sense. But the limitation is the mindset of the people.
Got it. I asked a question. This was for Georg originally, but I think it could go for anyone. A thing I've been thinking about was there are many different methods to measure MRS, to do the quantification part. And it seems like there isn't a consensus on what's best. Otherwise, everyone would use what's best. Is there any benefit of using many of them and doing some sort of ensemble like measurement?
Yeah,
and maybe for other quantitative.
I love the idea, and I responded already in the chat, but it's that was at the heart of a grand application that I put in. It's like some similar work has been proposed for fMRI by Greg Keir in New York, where they face similar problems, right? Like if you drag the same data set through like 20 different pipelines, you get like non-congruent results. And we face the same thing. So why not like drag it through all of them and like run a little meta-analysis on it? And I really like the idea. I think it would be complementary to this synthetic validation approach that I was talking about earlier. But I generally like the idea. We have a finite number of algorithms. Within the algorithms, the other complicating like dimension, I guess, is it also depends totally on like the kinds of settings that you use, because they all can be customized a little bit. So like you have a you have a multidimensional space really of analysis tools and analysis settings. But you know, I totally like the idea, and I want to pursue it further. Of pretty much what you propose, literally taking the same data set and just punching it through 100 different pipelines, for sure, and then just seeing what sticks.
Cool. General question for the group. Are there any practices you've seen from each other's talks that you might be able to implement in your own work?
I can start. I was super excited to listen to Georg's talk, because literally today I was looking at how to implement Osprey for my own MRS data. So yeah, I will. On the flip side,
get in touch. get in touch. I'm sure we can make that work. Like it shouldn't be too hard.
Yeah. On the flip side, I think for the animal part, at the moment, there's no benchmark yet or there's no need to really say like the basics parameter like T1 or something are the same across centers, because we're not coordinating large clinical trials which really rely on this thing. So we don't necessarily have that drive. That's the other side of the medal for my personal work.
And that's fine. I think like in preclinical animal stuff, there's still like in humans, there's not a lot of normative data to like streamline the quantification and so on. My guess is there's even less in rodents or in mice. The upside is you can be the person to kind of come up with them, right?
Yeah, I mean, eventually, going back, I think with some thoughts that we have in the animal community is, try to have something big put together now that we've gathered a critical mass, and there will be a few applications where we actually can't just do all the recordings in one single lab. So then the moment we start thinking about that, then we need to start thinking about making sure that our MRI's machines detect the same things. We have the advantage that 90% of all of the systems come from the same vendor. So theoretically, we hope that we can remove some of the problems that we've seen, like for instance from Georg's data sets, where we see, was it Siemens that was having most variability in the spectroscopy?
They all do. It wasn't particularly specific to Siemens. Like
okay.
They don't, maybe that was like a visual artifact, but I'd see within vendor you see similar kind of like side variances.
Yeah. So that's the one strength that we have, is that we have one big vendor. It's also the weakness that we have.
I'd argue it's more of a strength.
Todor, what has been one of the most challenging steps for producing reproducible quantitative phantoms?
Sure, reproducible quantitative. Yes. So, well, challenge is first of all that all these phantoms should not have any magnetic component. Sure, they should not have any metal component. So you have to control all the properties of the vessels that you're using. Then you have to make sure that you measure the properties at many different field strengths, and that could be challenging, since we would like our phantoms to be measured, most of the mimics to be measured at NIST if possible, and standardized this way. We have, of course, in-house measurement capabilities, but they cannot compare to what NIST has. So that's kind of the most the other challenge is, of course, the long-term stability of all the mimics, all the reference objects. So you have to keep track of that. And at some point, you have to define the lifetime of these solutions that we're using, which is currently at about 3 years. And of course, development of new tissue like mimics is it's extremely important. And here I could say that these tissue-like mimics could have both MRI properties, including MRS properties, including magnetization transfer properties, including CEST properties. So all this, it's kind of challenging for developing new mimics, but we are working on that too.
That segues into another question. I was wondering, you mentioned the actions that you take when measurements are off by a threshold. And I was kind of wondering, I mean, ideally the standard objects wouldn't change over time or be altered or dropped or something if you have like a geometric thing. Is there a way that you can test them independently from the system, or so that you know that it's the system that's changing and not the phantom object itself or the standard object itself?
Sure. So the standard objects, that's why we have an array of mimics. So for example, if you'd like to measure the apparent diffusion coefficient, you're not going to measure in a single point. So we have very different solutions, starting from what you have in apparent diffusion coefficient in water, and then to the extreme case of extreme pathologies, and even going beyond that. So if you see that consistently only one or two mimics are off and all the rest is fine in the mid-range, for example, so most likely something is going on with the physical object. But if there is a shift, and that's, you can easily see this when you do longitudinal study, if there is this constant drift of the parameters over time, so of all of them, so something, this could be coming both from the scanner side and from the phantom. So we have to definitely think about that, and of course send another reference object just to make sure that it's not the first one that is causing the problem. And
that's a good point.
I have a question to Georg. So, I mean, to both of you guys, how you calibrate your techniques? I mean, fMRI, we know there is no calibration there. All two people claim that. But how you calibrate your MRS?
Define calibration.
So let's say I'm looking for nanogram per milliliter of a given substance.
Oh, okay. Yeah. And I think I see where you're coming from. So yeah, that's a super complicated issue that gets the more the more you look into it, the more complicated it gets, right? So what we ultimately get are these spectra, which we put through a model and we can kind of decompose it. But if you wanted to, or if you want to translate them into like measures of concentration, that is really hard. Like there are essentially in human brain spectroscopy or in animal brain spectroscopy, there's usually two different ways of doing your estimation. One is just to assume that total creatine is constant across people and across brain regions and across tissue and so on, and use total creatine as an internal reference. So you say like, I'm getting NAA to total creatine. That is something that is totally being done. It obviously completely papers over the fact and ignores the fact that creatine might not be constant. And in fact, there's a mountain of evidence now suggesting that for example, it changes with age, certainly changes with pathology. So things like that are difficult. The other way that we can do it and that we can actually get close to concentrations is by normalizing to the water signal, right? We can measure the water signal, and we have a pretty good idea, at least in healthy tissue, how much water there is in gray matter and in white matter and in CSF, right? So what we typically do is we co-register our voxel to a T1. We do a segmentation, and we know the relative tissue fractions of gray matter, white matter, and CSF. And then we do a bunch of assumptions on relaxation correction. And that is really where it becomes problematic, particularly with like interindividual differences in pathology, is we're making assumptions based on literature values for these relaxation times. And so in that sense, you might argue, well, then you're not really calibrating it because you're making all these assumptions, and you would be right. Other people use things like, they calibrate against a phantom, but that is super hard to do because everything changes in a phantom, relaxation times and so on. So that just becomes also a non-starter. So ultimately, it's a lot of educated guesswork, and you arrive at things that resemble concentrations, but you have no way of knowing that they well and truly are. We're trying to get as close as possible to getting there, but that's a fundamental, instrumental and kind of physical limitation, is that we aren't directly able. We always have to be relative to another signal, so to say. Does that make sense? Stunned silence. Awesome.
I think it makes sense. Joe, I don't know if you had something to add there, or I could also have a question about clarity on the better protocol and how it was selected.
The point is, as Todor said, for fMRI, I think there's no, people claim calibrations. I'm not even going to go into that debate. But yeah, for the protocol, I went quickly over this purposefully because it's tedious details in the end. But we had a few hundred scans, as I said, and so we looked for what were the commonalities in the good scan compared to the bad scans, and so from that we got an idea about anesthesia and sort of kind of SNR range that we needed to be. And then actually I called everyone up and I said, well, we're going to have discussions about this new protocol. And I was expecting that this would be a fight, because, we're all from the same community and we love to fight about protocols. And there was none. I just said, look, guys, this is what I think seems to work. Do you want to change anything to it? And I said, no, it's great. Let's do it. And they agreed. In less than an hour, we had a protocol. Wow. Which I didn't believe. So it is possible to get 50 people in a room to agree on a protocol eventually.
That's amazing. That's great.
There's hope.
Can you do that for the spectroscopists too? I'll pay you.
Yeah, that seems like we could all use that. Okay, I think with that we can wrap up the session. Thank you again to all the speakers for the great talks. That was awesome. Last point of reminder is that there are many other sessions in the conference. There's one every 5 hours, so feel free to look at the program for anything of interest to you. And yeah, thanks again.